Good morning, everybody. All right, let's get it cracking. Okay, so today we're going to be finishing up transcription. Good morning, Rogelio, and everyone else. And let's get that up. Here we go. So this is pretty much where we finished up on Friday. And so again, kind of continuing the theme of bacteria a little bit simpler in some ways than eukaryotes. Essentially, now we're going to go to look at eukaryotes, and things get a little bit trickier. So, in large part, hang on, where did that? Oh, I've already gone over the gene structure. Oh, okay, good. Just refreshing my memory. If you remember back to where we talked about eukaryotic genome structure and the like, which was back up here. And where the hell's the promoted part? Ah, well, this is the problem of using someone else's stuff. It's always hard to figure out what the hell is where. Okay, so anyway, you remember from earlier when we talked about eukaryotic genome structure, right? We have a basically a core promoter in eukaryotes, right? So that contains all the stuff that the kind of transcription apparatus binds to, the holoenzymes, all the various transcription factors and the like. And then we have what's called a regulatory promoter, which is potentially far, far longer, right? So the core promoter can be, you know, maybe 150, 200 base pairs long. And this is the absolute required bit which is needed for transcription of that gene, right? So if you stick that in front of another gene, you'll get transcription at a certain level, right? Not necessarily very high, it's not going to be controlled in any particular way, but it will, you know, kind of chunter on its own, right? And so this is controlled, or how effectively this works is controlled by the regulatory promoter, right? Which contains a whole bunch of sequences that are recognized by other proteins called transcription factors, transcriptional activator proteins, repressors, all kinds of jazz, right? And so this is where it gets really kind of, well, I'd say, batshit crazy, right? And that's where an awful lot of the complexity in control of expression in eukaryotes comes from, right? It's this regulatory promoter over here. And this can be huge, right? Thousands of base pairs long, right? And in fact, some parts of it that you can see over here can be literally millions of base pairs away, right? On the other end of the chromosome. In fact, there's one of the genes which is responsible for polydactyly. You get kind of extra fingers coming out of your hand over here, is because one of these long distance regulatory elements is mutated, right? So it doesn't work the way it's supposed to. Okay, so essentially in that, where we're at, here we go. in that core promoter, you now have a Tata box, right? So remember in prokaryotes, sorry, we have a Tata box, a -10 and a -35 sequence. These are responsible for RNA polymerase binding and also the region which is kind of popped open by the RNA polymerase when it bends the DNA, which you can see just up here. See how it kind of bends the DNA in a right angle, and in the process forces those two strands apart. So in eukaryotes, there's just a Tata box, right? And so the core promoter sequences in eukaryotes are much less conserved, right? And so these are a whole bunch of different genes. I don't even know what any of those are, to be honest. And you can find about 25 to 30 nucleotides kind of upstream of the transcriptional start site, which is here. You'll find a largely T and A box, right, called the Tata box. And as with prokaryotes, how close to that consensus you get can affect how strong that core promoter is. Stop me at any time too if there's anything you want clarifying. More than happy to. It'd be nice to hear some voices. So there's 6 general purpose transcription factors, right, which work with RNA polymerase 2. Remember, RNA polymerase 2 is what transcribes mRNAs. Where did we have that? It's up here somewhere. Here we go. Right. And also some SNRPs as well, which we'll get to later in RNA processing. Remember, RNA polymerase 1 is ribosomal, RNA polymerase 3 is tRNA and some other ribosomal stuff. Okay, so essentially we have some general purpose transcription factors which facilitate RNA polymerase activity, right? And so this could be by recruiting RNA polymerase to the promoter, could be by stopping it from leaving the promoter, right? And so these are just kind of general purpose ones, right? These aren't necessarily ones which change in response to specific conditions, right? So these aren't ones which will kind of turn those dials, right, that we're talking about. These are just needed for RNA polymerase to work. And so you can see the whole complexity going on here, right? So the very first process is we have TF2D and a protein called TBP, which stands for Tata box binding protein, or Tata binding protein. Or maybe Tata box is one word, I don't know. Anyway, T and a B and P. So this recognizes the Tata box, right? That sequence that you can see here in the core promoter. And it serves as a like, hello, for RNA polymerase to assemble around. It also tells RNA polymerase where it should bind, right? And that's kind of done differently in prokaryotes, right? That's more sequence specific in prokaryotes. In eukaryotes, it's based on the actions of this little complex of proteins of the transcription factor, the TBP. Otherwise, RNA polymerase doesn't know where it's going, right? It could be like anywhere in here, and that would not be good. Okay, so once the TBP and the TF2D have recognized that Tata box, then all this other jazz assembles, right? We have our RNA polymerase holoenzyme. We have all these other transcription factors, right, which are in part responsible for either recognizing this protein or recognizing bits of DNA in the promoter and kind of keeping RNA polymerase there. And there's also another protein called mediator, right? A mediator, as the name suggests, mediates an interaction between RNA polymerase and other regulatory elements, right? This is essentially the switch that is flicked, right, to start transcription. And so all of these assemble together, right? And this can be interrupted too. This is what we'll get to when we get to control of gene expression. Any proteins that bind in this core promoter can interfere with this assembly process. Right, so if you want to shut down gene expression, blocking the binding of any of these will do that. Right, so that's one way you can stop it. So then we have our regulatory promoter. Ooh. I'm sorry. Which, again, can be of hugely varying length. It's actually very hard to pin it down, to be honest. And we have other proteins called transcriptional activator proteins, repressor proteins, all kinds of other proteins, right, which will bind to specific DNA sequences in this regulatory promoter. Right, so these DNA sequences are recognized typically by just one protein. Right, so if you have that sequence in front of a particular gene, it can be activated or repressed by that protein. If you don't have it there, you can't, right? So this is how we get a lot of the complexity of control of gene expression. That's one reason why we are more complex, right? It's not just the genes that we have and the DNA we have, it's how we use them, right? And there are an awful lot of different ways in which you can turn a gene on or off at many different levels. So anyway, these transcriptional activator proteins, and again, this is a fairly simple version of what's going on, believe it or not, kind of bend the DNA, right? And so they cause the DNA on the actual chromosomes kind of bend round. And that brings another activator protein in proximity to the mediator protein. And as soon as this touches, off it goes. Right, away are the races, so to speak. It's like a greyhound that's chasing a rabbit. Actually, my dad has just adopted a greyhound, which is kind of neat. So anyway, so this whole complex just sits there, right? Sits there waiting for this to happen. If it doesn't get activated, it doesn't transcribe, or at least not very well, right? It might be able to do so at a low level, but it's not going to be particularly efficient. Right? Remember that core promoter will work, it's just not going to work at a very high level necessarily. Anybody have any questions? Questions. Okay, so essentially once that happens, elongation, the bit where the mRNA is made, is fairly straightforward. The RNA polymerase just zips along, incorporating RNA nucleotides into the ground strand. That bubble, get out of it, there we go. That bubble that we're talking about that's created by RNA polymerase, and this is the same in both prokaryotes and eukaryotes, zips along, and eventually it's got to stop, right? And so there's nothing very much interesting going on with elongation. It's simply the production of an mRNA transcript. However, termination of transcription is where things get interesting, because one has to stop, right? You cannot continue this process all the way along the chromosome, because A, you'll produce transcripts that you don't want, and B, it'd just be a huge waste of resources, right? You know, if you can imagine how much energy goes into this process of making the bits involved in it, yeah, not a good idea. Right, so we've got to stop this somehow, right? And so there are essentially three different approaches: two for prokaryotes, and one for eukaryotes. And I'm going to go through each of those in some detail to kind of give you an idea of how this can happen, right? And so in bacteria, there's two main mechanisms, right? There's intrinsic termination, right? And essentially what that means is that there's something in the actual mRNA sequence, or the DNA sequence as well, right? But in the DNA sequence, that when it's transcribed into mRNA, stops transcription, right? That's called intrinsic. Whereas there's or the opposite obviously would be extrinsic, right? Something from the outside. And that's called row-dependent termination. Row is the name of the protein, right? And so again, there are some features in the mRNA, but then they don't terminate transcription themselves. They actually facilitate the action of row in termination transcription, right? And so one thing I want to get across is that particularly in bacteria, there's a really huge role for secondary structures of mRNA, right? And so that's something that really is worth remembering, right? So we've talked about secondary structures of RNA, right? Yes, I think so. Yeah, get back there. Right, so these kind of loops and all kinds of funky structures and things like that, these are really important in a lot of bacterial gene expression. And we'll be coming back to these in control of gene expression in prokaryotes. Right, so kind of keep that, in your mind. Okay, so essentially what intrinsic termination is all about is that there are two kind of main regions. There's one which is a string of A's in the DNA sequence, right? When that's transcribed into uracils, for some reason, RNA polymerase kind of stutters and pauses over uracils. For some reason, if there's enough of them. I don't actually know why. The other thing is there's essentially, like that, right? An inverted repeat in the mRNA. What an inverted repeat means, actually it's probably better to do like that. There's two sequences that are the same, or the opposite, but in facing each other. So if you remember the same kind of thing that you find in transposons, it actually does a very similar kind of purpose. And so when that inverted repeat is transcribed, those bases are able to base pair. So that kind of forms a loop. If you can imagine, like there's a little loop on the top of my fingers. Limit, I only have like a certain number of joints on my fingers, right? And so once that's transcribed, that loop forms, and that loop actually forms inside the RNA polymerase in part as it's being made. And it kind of just boots it off, right? And so if you want to look at it in kind of pictorial format, here's our inverted repeats, right? Here's our string of A's, right, which we transcribed into U's. And so essentially RNA polymerase is going to start here. It's going to chunt along, kind of through here. It's going to make this pair of inverted repeats and pause just here, right? And so when it's paused on these U's, this inverted repeat kind of bonds together, right? So if you look at it here, essentially you have going this way, AAA, GG, CT, CC. And at the top, going the other way, you have AAA, GG, CT, CT, CC, right? So you have the complete opposite, right? Kind of going like that, which means that when you transcribe just one strand, those two will base pair and form together. And there's always a few bases in the middle to create this kind of loop structure here, which is what allows that mRNA to bend and form double-stranded RNA. And if you imagine that RNA polymerase is actually here, and it's obviously it's a huge enzyme, right? It's a massive complex. This physically kind of pushes it away from the DNA that it's sitting on, and it falls off, falls to pieces. Ta-da! Just like that. Now, row-dependent termination is different, and there's actually a little bit of uncertainty about actually how exactly this works. So I'm not going to get too much into that because I don't understand it well enough myself. I just read a paper or a kind of commentary about a paper in Science a few weeks ago about how they might have figured out how this actually works, but it's like whoosh, straight into my head. Okay, so essentially row-dependent termination uses a protein called row, right? I don't know if that was, actually I have no idea where that comes from. I don't know if that person who discovered it or people who discovered it had like a nursery rhyme stuck in their head, like row, row, row your boat, maybe. I don't know. Anyway, whatever reason, it's called row. And it essentially has, again, another sort of feature which causes a pause in transcription, right? And so the whole deal with row-dependent termination is that row is trying to catch up with RNA polymerase. And so when it does, it kicks RNA polymerase off. And so essentially, row is an mRNA binding protein, right? So it binds to RNA. The RNA polymerase pauses, right, because it's hit, a series of U's maybe. I'd rather see what it says in the picture. And then there's no secondary structures in the RNA, right, which will slow row down, basically. And so row is kind of basically just like running along behind RNA polymerase, trying to catch up with it. RNA polymerase pauses, and if I could get my ass off the couch and show you, I'd be like, boots it off with my foot. But obviously I can't do that because I have my laptop on my lap. Anyway, so that's the general mechanism, right? Here's what it looks like in a little bit more detail, right? So again, you have this row binding site, right? So you have a stretch of RNA encoded by the gene in the DNA, which are recognized by row. RNA polymerase encounters a terminase sequence. Oh, actually, that is inverted loop. Forgot about that. And that slows it down. It's not enough to kick it off, but it slows it down enough for row to catch up. And then when row catches up, it kicks off the RNA polymerase using, essentially by forcing apart the DNA RNA hybrid. Right, so if you remember all the way back here, right, RNA polymerase has within it an RNA DNA hybrid. The RNA that it's making is base paired, at least for a short stretch, to the DNA that it's copying, right, the template strand. And so row is going to come in here and it's going to literally kind of unwind or force apart that hybrid. And so the mRNA leaves, and then the RNA polymerase basically goes, oh, fair enough, I'm done here. And it leaves too. So again, it's a matter of speed, right? So that row protein can process along this mRNA at a certain speed. And if the RNA polymerase never stops, it will never catch up and stop transcription, right? And so that's why RNA polymerase doesn't get stopped before it should be. And then we have essentially the same kind of deal here, this intrinsic termination loop. We'll talk about those a whole bunch more in the next section. This intrinsic termination loop, which doesn't kick the RNA polymerase off, but slows it down enough that allows row to catch up. And that's a different way of doing it. I don't actually know why, again, I don't know everything. Actually, I'm pretty happy that I don't know everything because that means I still have stuff to learn, right? But I don't actually know why you would find one of these mechanisms over the other in any given prokaryotic gene. I don't know if it's more common in one type of bacteria than another, or one type of gene or another, or if it's just random. I don't know. That's something I'll have to look into, see if I can figure that answer out. Actually, well, I would write on the back of my hand, but I can't find where my pen is gone. My wife's tied it away. Okay, so in eukaryotes, obviously, they do something different, but it's the same kind of principle. So here, there is no real kind of sequence that tells the RNA polymerase to stop or slow down. But we have the same concept of something catching up with RNA polymerase to kick it off, right? So same kind of deal as Rho, but a different mechanism, right? So RNA polymerase isn't slowed down like it is here, but there is still a protein that's trying to catch up with RNA polymerase. And so this protein is called RAT1. Yeah, I don't know why it's called that. And what RAT1 is an, it would be an endoRNase, right? So it would be an endonuclease, something that cuts within a DNA or RNA sequence. Let's see if I can get chat. Oh, messages. Sorry, Sophia, it was, oh yes, we have in referencing. You have questions?
No, that was to the RNA secondary structure.
Oh, okay, great. I'm sorry. It doesn't give me a timestamp, so I've got no idea when you said that if I missed it.
No, it's okay.
My box has a life of its own for some reason. Okay, so yeah, thanks for that, Sophia. So an endonuclease cuts within a DNA or RNA sequence. Here you go. Who can remember an exonuclease? Or a protein that has exonuclease activity? This is kind of make you think a bit hard about stuff that we've done maybe a little while ago. It's actually part of a much bigger protein. Stops when it makes a mistake and goes back and cuts that base off. You remember? Really important in proofreading. What am I talking about? Yeah, DNA polymerase is exactly right. Thank you, Jane. So an exonuclease can cut only the end. And they also are specific to ends as well. So 5' versus oh, that's a terrible bit of writing. Right, so that's why once DNA polymerase has moved on, it can't then go back and cut out the mistake that it made if it has already added another base after that, right? Because that would require endonuclease activity, which it does not have, right? And so this is actually really important because when we get to talk about RNA stability, particularly in eukaryotes, right? So how long mRNA hangs around for, protecting it from exonucleases is absolutely critical, right? And actually opening it up for exonuclease activity is one way which we get rid of mRNAs that we no longer need, right? So one way of shutting down gene expression. So anyway, that's a little bit of a tangent, but it's worth kind of keeping in mind. I try and kind of like drop little breadcrumbs as I go through that we'll pick up later on so that when you see stuff later on, it's not the first time that you can encounter it. So that's why I talk about this stuff. It's not that I'm like trying to tease you with all this cool stuff that we're going to learn later. It's more so that when we do get to it, you already have some preparation for that. Anyway, so RAT1 is an endonuclease, and it's an RNase, right? So an RNase degrades, as you'd expect, RNA. Ta-da! And we actually have RNase to the nth degree all over us, right? From bacteria and other places. I don't know what. But that's why whenever you work with RNA, you have to be super, 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 super careful not to introduce any of those RNAs into your sample. Thankfully, we're not going to be working with RNA in genetics lab because it is a royal pain in the ass. But if you did, you'd be like gloved up. You'd almost be wearing like a hazmat suit, right? Not to protect you, but to protect your sample. So mRNA is very fragile. Anyway, so this endonuclease, what it does is it recognizes this cleavage site. The RNA polymerase is blissfully unaware that this has happened. It just keeps chugging on, and RAT1 cuts this bit of pre-mRNA. It's called pre-mRNA because it's not actually mRNA yet. We'll get to that when we talk about RNA processing. Now, once it's cut that bit of pre-mRNA, this piece of pre-mRNA is still actually technically not mRNA yet. Yeah, a little bit wrong with that. This pre-mRNA can go off and be processed, right? Processed, get all the jazz added to it that it needs to, and then it gets hoofed out of the nucleus into the cytoplasm, right? And in the process, RNA polymerase just keeps on going, right? Blissfully unaware. It would just keep on going until it fell off the end of the chromosome, pretty much. And so now it's the job of RAT1 to catch up with it and stop it, right? So it's like this race against time. And I don't know. I've, with my dad when I was growing up, watched so many car chase movies. It's kind of like that, I guess. So the way it does that is by digesting this kind of, I don't know, unneeded bit of mRNA that's being made by RNA polymerase. And essentially, it's a battle between how fast can RNA polymerase make it and how fast can RAT1 destroy it. This also has the kind of neat side effect of, in the process, recycling that RNA, the RNA nucleotides, which can then be used again, to make more RNA that's actually needed. Right? And so RAT1's attached, it's going to degrade the RNA as it moves towards, so it has a processivity, right? Kind of like Pac-Man, going to chews up the mRNA and keeps going essentially until it reaches the RNA polymerase. And when it does so, I'm not entirely sure about how it does this, to be completely honest, but it essentially kind of gets in the space, partly into that channel where RNA polymerase is bound to DNA and mRNA, and in the process continues chewing up the RNA and kind of levers off RNA polymerase from the DNA in the process. Now, again, remember, this isn't a one-shot deal. It depends on how strong that promoter is. But technically speaking, as soon as this RNA polymerase has cleared this region, another RNA polymerase complex can assemble and start transcription. Right, so this is a continuous process, obviously depending on how strong this promoter is. If it's a fairly weak promoter, maybe that's not going to happen so much. If it's a very strong promoter, I'm trying to think of a really good example. Probably be something like if you had some kind of muscle damage from an injury or from doing some strenuous exercise, and you needed a lot of myosin filaments, right, to repair that. And myosin and actin are the the sliding filaments that contract your muscles, essentially. Then you'd want an absolute ton of it produced very quickly. And so you'd kick expression into high gear. You'd have a very strong gene expression of myosin until you didn't need it anymore. And then you'd shut it off, basically. So while this is happening, there is going to be another RNA polymerase potentially over here trundling along and making an mRNA as well. Right? It just depends on the promoter and the gene itself. Okay, so anybody got any questions on any of this stuff? Again, we're going into quite a lot of detail here, so one would rationally expect quite a lot of detail in the exam, and particularly on, you know, when I'm talking about this speed issue, right? You know, so this is the same as in row, right? We have to slow down RNA polymerase for row to catch up and force it off. And exactly the same principle applies to RAT1 termination in eukaryotes, right? In this case, we're not actually slowing down RNA polymerase, but the thing which is catching up is faster, and so it catches up, right? But at the end of the day, what you have is a protein that interacts with RNA polymerase and the RNA and the DNA and forces it off, right? So the principle is the same. And you know that I'm big on kind of principles and concepts and stuff. So that's also something that's almost certainly going to be in the exam. All right, so next, and we won't get through all of this at all, right? Because this will take a whole bunch of time. So actually, we're way, way ahead of time. We might even have time for doing some recap type stuff before the exam. Maybe talk about some other funky things. Okay, so essentially what we've done, at least in part, is this bit, right? So we've done the, sorry. We've done the transcription part, but we still don't have functional mRNA yet. This is a real key point. There's an awful lot that goes into getting the mRNA or the pre-mRNA prepared and turning it into mRNA. So there's kind of three things that need doing. What's one thing that needs doing to the pre-mRNA that's just been transcribed? Think about the eukaryotic gene structure. What needs to be removed before that mRNA can be translated? Now think back. Are eukaryotic genes collinear or interrupted? Yeah, so good guess. I mean, you got a 50% chance of getting it right, I guess, but I'm going to assume that you said that because you know it. But what are the bits that are the interruptions? And the in is part of the name.
Okazaki fragment?
Oh, actually, no, that's a good, that was Jeremy, right?
Yes, that was me.
Fragments are generated during DNA replication in a lagging strand. What we're talking about here is the actual structure of the eukaryotic gene itself, not necessarily the DNA, but how it's organized. And Sophia got it in one. These are called introns. Yeah, let's go back up. Where are we at? Okay, so this is kind of, essentially, I try and do this so that this is almost like part of a revision of the material, right? because going back over this stuff is how this stuff gets buried, kind of, handed into your brain. And so essentially eukaryote genes are interrupted because they have these things called introns. These introns are non-coding pieces of mRNA, i.e. they don't code for protein. The only bits that do are these exons, bits in green. And again, these aren't like physical structures in the gene or in the mRNA. These are simply kind of representing different functional blocks, I guess, right? Kind of cartoon style. So these have to be removed, right? Absolutely critical. If these are not removed, and actually there are a class of mutations called splice junction mutations, which will make a little bit more sense once we've gone through it, that if you have mutations in the splice junctions, right, which are used to get these out, you almost always generate a funky truncated protein, which is non-functional, because translation will continue into an intron, which it shouldn't, and then it'll stop because there'll be a stop codon in there, right? So we need to get these out. Also, where you have a poly A signal here, and you're like, well, that's a poly A signal, right? So we'll also go into what that's involved. Good morning, love. And so there's kind of two main purposes to RNA processing in eukaryotes. One, obviously, is to remove these introns. And two is to protect the ends, right? So that if you remember when we were talking about exonucleases, there are an awful lot of RNases, which are exonucleases. So if they find an unprotected end, either the 3' end or the 5' end of this transcript, they will start chewing up the mRNA, and then all of your efforts in producing it are going to be wasted. So the other part of RNA processing is about protecting these ends. Oh, doctor come down from bed as well, maybe 10 o'clock. Okay, so those are two separate processes, the 5' end and the 3' end, but they serve the same purpose. Right? So again, removing the introns, protecting the ends. Those are the two big deals in mRNA processing. And obviously, because only eukaryotes have these, this is something that only happens in eukaryotes. You don't have any mRNA processing that I'm aware of in prokaryotes. And actually, prokaryotes are all kinds of funky, really. I mean, I guess if you're a prokaryote, you're like, well, eukaryotes are funky, right? Depending on your perspective. But being someone that's worked with eukaryotes for the vast majority of his career, bacteria always seem a little bit odd. Nothing against them personally, obviously. Okay, so all of this happens kind of as transcription is going on, right? Actually, almost as soon as transcription begins. And so the very first thing that's required is the addition of what's called a 5' cap. And so basically, as soon as, or do I have a better picture? No, I don't. All right, as soon as this part of the transcript is produced, literally as soon as it emerges from the RNA polymerase, right? So once this whole complex is moved kind of over here, and you have a little bit of mRNA popping out, which is the very beginning of the gene here, or not the gene, the open reading frame, I guess. As soon as that's produced, you have to stick a cap on it, because otherwise it'll be recognized by exonucleases, which will just destroy it essentially as quickly as it's produced, which will be super annoying. Okay, and so this serves two purposes. One, to protect it, which is what we've talked about. But actually in eukaryotes, this is also involved in ribosome binding. right? So once this mRNA is processed and exported into the cytoplasm, there has to be some way for a ribosome to recognize it and begin the process of translation. And this 5' cap, and it's called a 7-mpg cap for this methylguanine, right? So as soon as the mRNA transcript emerges from the RNA polymerase, this cap gets added. And this is kind of what it looks like, right? So we have the very first, I don't know where, this is kind of where it would be added to, like the very first base on the transcript. You get this kind of triple, oh, I don't want to go deep to that. Sorry. This is telling me I got stuff for 15 minutes. This is how I remember things. This basically methylated trinucleotide group gets added onto the 5' end of the transcript. Now, you probably are thinking, well, hang on, you've always told us that you can only add nucleotides onto the 3' end. Well, this would be the exception, essentially, right? And once this is on that end, essentially, RNases or exonucleases don't recognize it. They can't bind to it and start chewing it up because it doesn't look like a normal RNA nucleotide for various reasons. Okay, so that's one thing, right? And so now that 5' end is protected, it'll start getting kind of, squeezed out of the RNA polymerase like toothpaste, I guess. And now you're going to start producing, actually, I don't know why this is at that point. Sorry, I got to put this over at this. And this is what happens when you use other people's lectures. They always have some kind of other way of talking about stuff, which doesn't necessarily make sense. Right, so the next point in the process is splicing. And so I always use an analogy that this is like in the old-timey, actually they still do it this way, I think, sometimes, when you produce like a cinema film, right? So like a reel of film that you're going to play a film on in the cinema, the whole kind of, I think there are still cinemas that use that. Obviously, it's all like digital now, I guess. But if you have a really long film, like, I don't know, Godfather or something, you can't fit it all on one reel. So you actually have to stick it together, and literally you use sellotape, right? Obviously, special type of sellotape, stick it together. And so it's like one continuous piece. Or if you want to edit a film, you cut out some scenes and then you stick it back together so it's one continuous piece of film. And that's essentially what we're doing here, where we have this long piece of information, and it has within it regions that we don't want, that cannot be there. Because if they are still there, they're going to cause all kinds of problems in translation. And so we have to get them out. And so the aim is to essentially kind of, if we have our two exons like so, make sure you can see on the camera, is to cut them here and here and stick them together so they're continuous. So it's no longer exon 1 and exon 2, it's just protein coding region, right? And that's the purpose and process of splicing. And so this takes the pre-mRNA, or primary transcript, get off of that. Here you go. It takes the pre-mRNA, which is produced by the RNA polymerase, and takes out those introns. And there's a whole load of involved in that, which we have no time to start whatsoever. So I'm going to call it a day at that point. If anyone has any questions, feel free to ask. Make sure I've got the chat up. and then we'll get into this next lecture. All right. Well, thank you very much, everybody. Again, if you do have any questions after going through this, just bring them to class, next class, and we'll spend some time going through them then. In the meantime, have a lovely day, and I will see you on Wednesday. Bye-bye.